first of all, take note that this is again the original replication for the eukaryotic chromosome. And what happens here is that there will be a replication bubble. And looking at the details of the replication bubble, you can see here that when the original replication separates from each other, then the DNA replication can actually proceed on one side and then proceed to another side, to the left side and then to the right side. Now, looking only at this box portion, you will be seeing here details of the DNA replication. So overall direction of replication can go to the left and go to the right, and there is what we call as the leading strand and also the lagging strand. We'll be talking about that a little bit later, but take note of here, this will be the direction of one of the daughter DNA or the complementary DNA being produced, while this will be the direction of the other complementary DNA strand being produced. So let's look at the details here. Now, looking at this picture, we will see here the different enzymes that are important for replication. First of all, you can see here the topoisomerase, over here is the helicase, and then the primase. We also see here the single-stranded binding proteins. Although we don't see here the DNA polymerase 1 and DNA polymerase 3 and also the ligase, but they will come into the picture later on in the process of replication. So first of all, there are two important here that are needed for the initiation or the start of replication. We first need here the helicase and also what we call as the topoisomerase. Now the helicase, the structure, the function of the helicase is that it is the one that will open up the double-stranded DNA so that single-stranded DNA can be produced in order for replication to happen. And how does it do it? Well, it does its job by breaking the hydrogen bonds between the two strands of DNA, and helicase will just continue to proceed until it is able to break down all of the hydrogen bonds between the two strands of DNA. So that will be the first enzyme that we have to take note of. This would be the helicase. On the other hand, there's another enzyme here called the topoisomerase, and it is very important because as helicase proceed to open up the double-stranded DNA to produce single-stranded DNA, it is possible, and that actually happens, that there will be a lot of entanglements that can happen here. And the job of the topoisomerase is to relax the DNA, the supercoils, and to remove the entanglements so that the helicase can proceed very smoothly into opening up the double-stranded DNA to produce your single-stranded DNA. So far we have discussed about the helicase, we discussed about the topoisomerase. The single-stranded binding proteins over here is also very important because its function is to prevent the single-stranded DNA that has already been opened up by the helicase to come back together and to re-anneal or to reform the hydrogen bonds, forming again the double-stranded DNA. So the single-strand binding protein will come into the picture, and then it will prevent the DNA, the single strands of the DNA, to come back together. So that's the topoisomerase, that's the helicase, and that's the single-strand binding protein. We'll go back to later to the other enzymes, but let's look at this one first. Here's another viewpoint of what the DNA helicase does. So here it breaks down the hydrogen bonds in between the A and the T and the C and the G. So in this case, the helicase will come into the picture, and then it will open up, remove the hydrogen bonds in between, and then simply producing single-stranded DNA and what we call as a replication fork, and that is how the DNA does its job, or the DNA helicase does its job. Okay, so let's continue on. Now that the helicase has already opened the DNA, producing double-stranded, single-stranded DNA, and the SSB has already prevented the single-strand DNA to come back together. Now, what comes into the picture is what we call as the primase, because the primase will be making what we call as an RNA primer. Please do take note that the DNA polymerase 3 cannot synthesize de novo. It cannot produce on its own a primer. So on its own, it cannot add nucleotides on its own. So there is a need for a primer in order for elongation to happen, because the RNA primer will provide the 3' end that will be needed to attach the nucleotides and to elongate the complementary strand. So this is what is very important here. So again, we have here the RNA primase going into the picture, and then it will add an RNA primer here, which will allow the DNA polymerase III to start synthesizing the leading strand. So there's a name here, leading strand. We'll go back to that a little bit later. So what happens next is that since the RNA primer has provided the 3' end, it will now it allows the DNA polymerase III to continue on adding DNA nucleotides in a 5' to 3' direction. And the reason it is called the leading strand, because it has a continuous elongation in the 5' to 3' direction, and then it's the same direction as the unwinding of the replication fork. So you only need one primer here, and then the whole complementary strand will continuously be produced, making it faster as compared to the other strand when it produces its complementary strand. So far so good. This would be the process wherein the leading strand will be producing the complementary strand. Now the next thing here is that we will be looking at the lagging strand. So let's look at the overview again. So here is the leading strand producing, elongating the complementary strand, adding the DNA nucleotides very fast, because it's only in one direction, or its direction is the same as the opening up of the helicase. So you can only have one complete production of the complementary strand. But the lagging strand over here would add DNA nucleotides on the opposite side, the opposite direction of the opening up of the helicase. So what will happen here is that the DNA polymerase needs to wait until there's enough space for it to add its nucleotide. So there is what we call as the addition of several primers by the RNA polymerase, so that there can be production of the complementary strand. So here is an RNA primer, another RNA primer, another RNA primer, and the direction of synthesis of the complementary strand would be on the other side, or would be on the other direction. So let's look at that in detail. So the lagging strand is opposite direction of the daughter strand synthesis, and in this case, if that would be the lagging strand, you have here a primer added by the primase. You have another primer here added by the primase, and later on there will be another primer here added by the primase. Once the primase has added the nucleotide, the RNA nucleotide, then the DNA polymerase can do its job, and then it will add DNA nucleotides at the 3' end of the primer, going in the opposite direction. So it's a 5' to 3' direction. It will go up to here until it completes the whole space, the whole gap, with DNA nucleotide. And this is called the Okazaki fragment. So the lagging strand will always be containing the Okazaki fragment. Let's look at that. So here is now the RNA primer. You have now produced the DNA nucleotide here, an Okazaki fragment. So this DNA polymerase will transfer again on the other side. Once the helicase has opened it up, another primer will be produced here, and the DNA polymerase will continue adding here more nucleotides. So another RNA primer, another Okazaki fragment produced here, and that is how the lagging strand is called lagging, because it's a little bit slower. You have to produce a lot of RNA primers, and then you have to have a lot of fragments over there. So once this is done, the DNA polymerase 1 will come into the picture, and then it will remove the RNA primer and replace it with DNA nucleotide, so that it will become pure DNA. So here the DNA polymerase 1 will remove it, as you can see down here, and then it will transfer over at this point, and then remove the RNA primer here with DNA nucleotides, completing the DNA complementary strand. Now the last thing that will happen here is that the gaps between the Okazaki fragments need to be sealed. So that is where the DNA ligase will be coming into the picture. Now in this picture, you will see here simply the demonstration, or the diagram showing how the nucleotides are produced. So here is the 3' end, the the carbon number 3 that is exposed here. So in this case, a nucleotide will add up here and it will form a phosphodiester bond between the phosphate and also the carbon 3 of the previous nucleotide, as you can see over here. So this would be the DNA polymerase job to add more DNA nucleotide. So this is the whole process, as you can see here, combining the leading strand and the lagging strand. So the leading strand only has one straight complementary strand because it goes at the elongation is at the same direction as the opening up of the DNA double strand into single strand by the helicase. But on the other hand, the lagging strand is in the opposite direction. So RNA primers are added as the helicase opens up because it has to wait before it is able to elongate, and the elongation would be on the opposite direction. Okay, here's a question. What are the bases of the designation of the daughter strand as leading and lagging strand? The answer to that is because the leading strand is faster, because the elongation is in the same direction as the helicase opens up the DNA, while the lagging strand is a much slower process because it will be going to the opposite direction, or the elongation is the opposite direction, which means it has to wait for the helicase to open up the DNA first before the DNA polymerase can elongate the DNA. Okay, so let's just finish up with this one by matching up the enzyme with the function. So you have the following enzymes: topoisomerase, DNA helicase, primase, DNA polymerase, DNA ligase. And what would be the function of this? So just look at this one, and let's start with topoisomerase. Among the 5 of the function, what will be the function of the topoisomerase? The function of the topoisomerase is it relaxes the supercoiled DNA. How about the DNA helicase? What's the job of the DNA helicase? It unwinds the double helix at the replication fork. The primase, its job is it the second, the third, or is it the fifth? It is the fifth. It provides the starting point for DNA polymerase to begin synthesis, and we know that this is called the primer. What's the job of the DNA polymerase? It would be to synthesize new DNA strand, also proofread and correct some errors. So the synthesis of DNA strand will be the job of DNase polymerase 3, while the proofreading and correction of errors, removal of the RNA, would be the job of DNA polymerase 1. DNA ligase, of course, would be to rejoin the 2 DNA strands into double helix and to join the Okazaki fragments of the lagging strands. And that will end our video for